All right, hello AP Bio students. I just want to do a quick recording of today's lecture on cell cycle regulation, which is chapter 12.3. So hopefully you find this helpful, but let's go ahead and get into it. Here are the topics that you should understand by the end of the lecture today. So the first question is, how does a cell cycle control system work? Okay, that's kind of like the major question of this concept. So specifically, how do cyclins and CDKs control the cell cycle in terms of timing? So what do they have to do with the timing of the cell cycle and when things occur? Also know about checkpoints and what the G0 phase is. What signals regulate the cell cycle? So there's external and internal signals that regulate the cell cycle, and then explain cancer and how that is related to the cell cycle control system. So let's go ahead and talk about the topics that are related to these questions. Now, when I was thinking more about the cell cycle control system, I was like, I recently watched ELF, and it reminds me a lot of how Santa's workshop works. So when you're running like a factory or a workshop, you have to regulate the amount of toys being made, the quality of the toys, when you start making the toys. And I was like, that's so similar to division of cells or the cell cycle. And so again, production of cells has to be regulated just like the production of toys also has to be regulated in Santa's workshop. So if you can imagine, how are they similar? Well, when elves are making toys for Christmas, they have to consider these things. Is it the season yet? Is it time for Christmas? Are we even in December? Because that's when we start making the toys, or at least a sense of urgency to make the toys starts to get higher, okay? So how do we know when to start making toys? That's the first question. How about the quality of the toys? When we're making the toys, how can we make sure that these toys are functional and they're not broken, okay? And because you can't give a broken toy to a kid, they're not gonna be happy, they'll be very sad, right? And so how do we ensure that the quality of the toys is good before we distribute it to the kids? Because otherwise, again, if it's broken, the function is not going to be there. And then finally, what toys do we need to make and how many of each toy do we need to make? And that depends on, the demand. So based off of your naughty and nice list, how many Nintendo Switches do we need to make? How many iPhones do we need to make? How many stuffed animals do we need to make? It just depends on the list. So how many kids want each of these toys? We don't want to make too many of one of them, okay? So let's go ahead and start with the first question. How do we know when it's the season to start making toys, or in the case of cells, when it's time to divide? So for Santa's elves, they know it's the time because they have a calendar. And so every year, right, come December, it's when they start making toys. So the calendar tells them when to begin preparing for Christmas, which comes every year on December 25th. So it's like a cyclical calendar, right? Every year we go January through December, back to January, all the way back to December. And so that calendar tells the elves when they can start preparing for Christmas. So in terms of our cell division, there's a cell cycle control system, which kind of acts like a calendar for our cells, okay? And it's basically a set of molecules in the cell that triggers key events and also coordinates these key events related to the cell cycle. It operates in a cyclical fashion, just like a calendar. Again, the calendar goes from January, February, March, April, May, June, July, August, September, October, November, December, all the way back to January. And so the cell cycle control system also works kind of like a cycle, okay? So G1, G2, or G1, S phase, G2, M phase, cycle over and over and over again. It's also regulated various checkpoints. And so we'll talk more about the regulation later on, but what's going on is at these checkpoints, it tells the cell, hey, you're not ready to move on to the next stage yet. Fix what's wrong before you move ahead. So it tells the cell to stop going on to the next stage, or it can tell the cell, looks like you're ready to go. Your quality is good, nothing's wrong with you. And so you can move on to the next checkpoint, you're ready. The major checkpoints are G1, G2, and the M phase checkpoints. And again, we'll talk more in depth about what happens during each of these later on in the lecture. So then in class, I had you answer a few slides, but we'll just talk about it together right now. So the two main regulatory molecules in the cell cycle control system that have to do the timing of the cell are protein kinases and cyclins. And so just to review, kinases are enzymes, proteins that activate other proteins by adding a phosphate group to them. In other words, they phosphorylate other proteins, which will activate those proteins, okay? Now, these kinases are actually always present in the cell. in equal concentration. So the concentration doesn't change. It's the same always in the cell, but it's not always active. Okay, so even though they're not activated until they attach to a cyclin. And the cyclin actually changes in concentration over time. So depending on what phase the cell is in, the cyclin will either have a high concentration, which will push the cell to the next stage, or it'll have a low concentration, which means the cell is not ready for the next stage. And these cyclins, again, they change their concentration in a cyclical fashion. So the cyclins kind of act like the calendar for the cell. It's like, okay, we have a lot, it's December, we gotta move on to the next stages of making toys. And then once those toys are made, the cyclin breaks down, and then you can rest and not worry about making toys anymore, or in this case, dividing the cell. So therefore, the presence of cyclin really is regulating the timing of the cell cycle. So the protein kinases are specifically known as CDKs, cyclin-dependent kinases, because their activity depends on the concentration of cyclins available in the cell. One specific one we're going to focus on is the maturation promoting factor, which is MPF. And that's really the first complex that was discovered. So let's talk a little bit more about this one. Another name for this is the end phase promoting factor because it moves the cell past the G2 checkpoint and says the cell is ready for division. And so first thing I want to note is the top left over here. I'm going to see if I can get my annotation tool going on. But this top left graph is showing us how the MPF activity in the orange over here, I'm just going to outline it in blue, is dependent on cyclin concentrations. As you can see, the cyclin concentration goes up first, and then the MPF starts to go up, right? So again, MPF is a complex of cyclins and kinases. And so when cyclin concentrations rise, then MPF starts to be activated, right? So again, cyclin-dependent kinases, that's what's showing us here. And specifically, you see that the MPF activity peaks during the M phase because when there's a lot of MPF, we're going through cell division. And so this is just another way to look at it. Down below, we have just a cyclical view of it. So as you can see, the CDK is always present. That's the orange, okay? The CDK is always present. However, the cyclin doesn't start popping up until maybe the beginning of G2. Okay, and that's when it starts to accumulate a little bit. That's where you see this region. So the accumulation of cyclins begins around G2. So that's like the upward part of this orange, I guess, curve on the in figure A. And then there's going to be a lot of cyclins available at that G2 checkpoint, which is going to push the cell into the M phase. Now, once the M phase is about done, the cyclin starts to degrade because we no longer want to keep the cell in the M phase anymore. And then once the degraded cyclin leaves, you repeat the cycle and you're back to the G1 phase. So let's talk more in depth about the specific roles of MPF. So in prophase, the MPF helps with chromosome condensation and spindle formation. So two of the major things that happen in prophase, MPF is helping with that. In prometaphase, MPF is phosphorylating proteins that is going to help to break down the nuclear envelope, which is, again, is one of the major steps in prometaphase. And then in anaphase, MPF is starting to turn itself off. So it destroys itself, as you can see right here. Okay, so this is really, oops, look at my drawing. This is really happening during anaphase. Because now we're about to finish cell division. We don't need MPF to be available anymore because MPF promotes mitosis, but if mitosis is finished, then we don't need MPF and we want it to break down so we can go back to G1. So obviously there's other cyclin CDK complexes, but we're just learning about one of them, and they're going to act maybe during other stages of the cell cycle. Okay, so not all of them play a role in pushing a cell to the end phase. They probably regulate the other steps, make sure things are good to go before they move on. So now we know that there's kind of a clock, right? Cells look at the concentration of cyclins to tell them when to move on to the next stage. But how else do cells know when to divide? You looked at this little experiment, and here's how this experiment works. So there's signaling molecules in the cytoplasm that can tell the cell to divide, okay? So here's an experiment where we see two cells. So I'm going to call one cell cell A, and I'm going to call one cell cell B. Give me 1 second. All right, we're back. Sorry, the light went off. So let's say that cell A is a cell in an earlier stage. So both of these cells are in different stages of the cell cycle. And cell B is at a later stage. They merged these cells and they noticed that cell A actually caught up to whatever stage cell B was at. So for example, in experiment 1, we see cell A over here is in G1. So I'm just going to label this. Okay, so here's cell A is in G1 and cell B is in the S phase. Once they're merged together, notice that cell A actually ends up in the S phase as well. And they even tested cell A being in G1 and cell B being in the M phase, which is much later. We're skipping basically the S phase and also the G2 phase, which is very important. But even combining these two cells pushes that cell from G1 to the M phase, even though it's not ready. So as you can see here, okay, the S phase didn't even happen. It's not even a replicated chromosome. So clearly there are some signaling molecules in the cytoplasm, molecules in general, that tell the cell when to divide. Let's learn more about these molecules, okay? So these molecules are going to tell the cell when to move on to the next stage. And so it's kind of like quality control. Is the cell ready? Is it high quality enough to move on to the next stage? Okay, you can't give a broken toy to kids during Christmas, and you can't have a broken cell move on to the next stage because it's not going to be a fully functional, healthy cell. And so quality control, if you remember from Buddy the Elf, he did not make enough toys. So he went to the quality control and he had to deal with all these Jack-in-the-Boxes, and it was terrifying for him because it just popped up at random points in time. But it's important that we send kids quality toys, and it's important that our cells are dividing into quality cells and they're fully functional. And so let's learn about the checkpoints that make sure that our cells are healthy and ready to move on to the next stage. Again, there's built-in checkpoints before G1, G2, and the M phase. And the G1 checkpoint is the most important one for dividing. Typically speaking, if a cell passes the G1 checkpoint, it's essentially set up for division. Okay, it's going to likely go to S phase, it's going to move on to the G2 phase, and then it's going to go to the M phase. If they don't pass the G1 check phase, the cell's going to go to something called G0, which is a non-dividing state. Okay, until some external signals tell the cell, hey, we actually need you to divide. And so depending on the body cell that you have, some cells stay in G0 for a long time and they stay very mature and they don't need to divide more. But other cells, like perhaps your skin cells, those ones, they need to divide very frequently, right? Because we exchange our, we lose a lot of skin cells every day. So we need to replenish them. And so these cells are likely just going to keep dividing pretty rapidly. But we have some cells in between that sometimes don't need to be activated, and at other times they do need to activate their dividing. So that's when G0 plays a role. When they're not dividing, they go to G0 until something tells them otherwise, we need more cells, and then they're going to go to the G1, or they're going to pass the G1 check phase into S phase. There's also the M checkpoint, and there's an internal signal during the M checkpoint that detects whether the sister chromatids are properly attached to the kinetochores before anaphase. And so this one's so important because we don't want to incorrectly divide our DNA, because that's going to really mess up our cell. Our DNA codes for proteins, and we want the proteins to be correctly coded for, because that's going to lead to proper functioning of an organism. And so if your cells don't divide the DNA properly, that leads to diseases and disorders and just malfunctioning. And so that's why we have this really important checkpoint at the M phase to make sure that our DNA is properly divided. Of course, there's more checkpoints, but these are the two major ones that we'll review. Finally, how do we know what toys we need to make and how many of each of these toys to make? Well, for Santa and the elves, they have a naughty and nice list that dictates how many of each toy needs to be made, right? Depending on how many kids you have on the list and what each of those kids requests for Christmas. And so, for example, maybe there's like 3 kids who want a Nintendo Switch, 4 kids for some reason who want Care Bears. This is a Share Bear, cute. And 2 kids who want some clothing, some Air Force Ones, okay? So the naughty and nice list is going to dictate, again, how many of each toy we need to make, because we don't want to make too many. If we only need 3 Nintendo Switches, we're not going to waste resources on making 10 of them, okay? If we need 4 Care Bears, we want to make sure we have 4 Care Bears. We want to just make 1 of them. We want to make sure every kid is going to get that cell that they need, or sorry, that toy that they need. And then Air Force Ones, again, we don't want to waste resources on making more than what's necessary. So then I had you work on this slide with your group, okay, and label different types of signals that tell the cell when to divide, but let's go ahead and talk about them. So there's internal and external signals that let our cells know how much that they need to divide. So the first one is a growth factor, okay? So growth factor is a protein released by one cell to other cells around it, and it's gonna tell them to divide. So one example is a growth factor called PDGF, okay? So platelet-derived growth factor. So platelets release this PDGF, which is gonna activate the division of fibroblasts, which are cells that are connective tissue cells. So basically, if you know, platelets are your cells that help with blood clotting. So let's say you get a cut or you have an open wound, platelets are going to basically start releasing this PDGF, which is going to cause the fibroblasts, these connective tissue cells, to start dividing so you can close up that wound. Okay, so that's what we're seeing in this diagram here. We're cutting human connective tissue, and we're going to add some PDGF. And we can see that with the PDGF, we start to see some cultured fibroblasts forming. But without the PDGF, then if there's no growth factor, the cells will not divide. So external signals are very important to causing other cells to start dividing. There's also anchorage dependence, which is that cells often need a surface to divide. Okay, so if they're not attached to a surface, they won't divide. And something, okay, so for example, if we go back to Santa's workshop, this is like a kid's name being on the nice list, okay? We need the kid's name to be on the nice list in order for a toy to be made for him or her. If there's not a name on that nice list, we're not gonna make that toy for the kid. Just like the cell, if the cell's not attached to a surface, that's not gonna divide. And then we need to know when to stop making the toys. So this is the density dependent inhibition. So cells, they stop dividing when they form one layer, okay? We don't need more cells. We finished already covering this surface. We finished making one layer. We don't want to make more. We don't want to waste resources making more. This is like making sufficient Nintendo Switches or Air Force Ones or Care Bears, whatever the toy is. We don't need to make more than necessary because otherwise we're wasting resources, okay? And we don't want to do that because there must be another part of our body that needs that energy more than if we already finished making a layer of cells. And so in this diagram here, we can see examples of both of these. So all the way on the left-hand side, we can see some anchorage dependence. So these cells right here, as you can see, they're in a Petri dish. They're in a Petri dish, they're attached to the plastic of the Petri dish, and that's gonna tell them to start dividing, okay? And as you can see, they form a layer. And so over here, they formed a layer and they've stopped dividing. This is density dependent inhibition. They form a layer, they're like, okay, we don't need to make more cells. And so they stop. But what happens when we remove some of those cells? Once we remove some of those cells, as you can see right here, they start to make some new cells. Okay, so again, we see that density dependent inhibition has been removed because the density has decreased. We have some open gaps until all that layer is filled again. And the density dependent inhibition kicks in once more, okay? So those are those some more signals that tell a cell when to divide, when they can divide, and when to stop dividing. Again, super important. So clearly timing, signaling, and regulation are very important for proper cell division and also function of Santa's workshop. Because without toy regulation, okay, without cell regulation, some evil elves might continue to make broken toys over and over and over and over again until they take over. And they're wasting resources, they're sucking up all of the good stuff from your body. And these are not good cells, they're not fully functional. These toys are not gonna bring joy to the children, and those broken cells in your body, if they're not regulated, they're not gonna bring joy to your body either. These are tumors, okay? And so I had you in class take a look at this meme. It says, what did the cancer cell say to the normal cell? To infinity and beyond. And I had you answer, how does this meme explain cancer? So let's talk about dysfunction in the cell cycle and how that leads to cancer. So cancer cells do not respond normally to regulation in different ways. So first of all, they might not even need to respond to growth factors. They just divide without growth factors, or they can make their own growth factors. They also have an abnormal cell cycle control system. So they probably just don't even listen to the checkpoints. They don't need the checkpoints to Like if the checkpoint normally says stop, they still go anyways, right? They're not ready to move on to the next stage, and yet they still go on. They can stop dividing at the wrong times, but more often than not, they just can divide indefinitely. So that's why it's like to infinity and beyond, because they just keep dividing, given adequate nutrients, even if their cell is messed up. Okay, and also, they don't die when they need to die because these cells are messed up, and usually messed up cells undergo apoptosis, but cancer cells, they don't. They just keep dividing even though they're broken. All right. So as you can see here in the top diagram, we see on the left-hand side, healthy cells. Okay, so we see anchorage dependence. The cells need to be on a surface in order to divide, and they are. And then once they divide and they form a layer, they stop dividing, okay, because of density-dependent inhibition. They form a single layer. Now, if you remove some cells, then that density-dependent inhibition is released, and so they fill that gap. And then they have another round of density-dependent inhibition once they fill the gap and they have that layer fully formed again. But if you take a look at the cancer cells, okay, they are forming multiple layers over here. So there's multiple layers. There are multiple layers. And also because they're forming multiple layers, technically some cells are not attached to a substratum. So they're not attached to a surface. They're just building on top of each other, accumulating on top of each other. And that's when we have a tumor. Abnormal growth. And we'll talk more about that and the different types on the next slide. Okay, so again, here's just another a zoomed up kind of image of normal cells versus the cancer cells. So if I was just to show you again, generally speaking, the normal cells would only form this bottom layer right here. As you can see, cancer cells, they don't show anchorage dependence because they're just building on top of each other. And so that's like no anchorage dependence. I'm just going to put AD. And then clearly, they also don't have density dependent inhibition because they formed a layer, or they're actually forming multiple layers now, they're not stopping. So let's talk more about some cancer terms. So in labs, when scientists are able to change normal cells to cancer cells that don't respond to cell regulation, this is known as transformation. Okay, so this is generally referring to cells grown in culture. So that's like in the lab. Now, in our bodies, when we have cells that have been transformed or cells that become cancer cells, they have abnormal cell growth. And when we have abnormal cell growth, that's an accumulation of these messed up broken cells on these cancerous cells. This is known as a tumor. And there's two different types of tumors. I'm sure you've maybe heard of these terms before, but there are benign tumors, and these are okay. I mean, they're never okay, but these are better than the next one. So in a benign tumor, the cancer stays at the original location and they can be removed by surgery. So that's what you hope for. If you ever have someone in your family or yourself affected by cancer, you hope it's benign, that's at a low stage, so that you can remove it by surgery. What you don't want is a malignant tumor, and that's one that invades other organs by moving through your bloodstream. And this is something known as metastasis. And so if you look at our diagrams here, okay, I'm gonna start the left-hand side. Let's say that there's a couple cells that start to become abnormal. Over here, it is becoming a tumor, but at this point, it's still localized. It's still just in one place. And so this is a benign tumor. However, if this tumor grows big enough and starts to invade into your tissue and start to access your blood vessels, that's when it becomes malignant, okay? Because now the messed up cells can send signals to other cells in your body to become cancer cells as well by invading your bloodstream. Because your bloodstream is connected to all parts of your body, and you don't want these cancer cells to be communicating with all parts of your body and causing this cancer to spread in that way. Similar idea here with breast cancer. So here is a small tumor, okay, from a single cancer cell. And it starts to get bigger in stage 2, starts to invade some neighboring tissue. And then over here, you can see that it's starting to metastasize. It's starting to get into your lymph nodes, and then it's going to also travel through your blood vessels. And that's what's going to lead to metastatic tumors that can travel to the rest of your body. And these ones are very difficult to remove because once they metastasize, you have no idea where this cancer is traveling to your body. And so what are some treatments for cancers that are metastatic? So there's high energy radiation in which your DNA is damaged So your obviously radiation is very damaging. That's why we wear sunscreen when we go outside. But cancer cells, they tend to be a little weaker than normal cells. And so they're more likely to be affected by high energy radiation. Of course, it's going to affect your normal cells as well, but because your normal cells are pretty robust and strong, they're not going to be as affected by the radiation as your cancer cells. So we're hoping to target most, if not all of your cancer cells with this high energy radiation without impacting your normal cells as much. And there's also chemotherapy. And so in chemotherapy, chemo is like chemicals. And so you're taking drugs that are toxic to dividing cells. They target cell division. So for example, there's Taxol. And what Taxol is doing is freezing the mitotic spindle. So as you know, the spindle plays a big role in dividing your DNA. And so if you freeze that mitotic spindle by preventing the microtubules from shortening, from breaking off those tubulin subunits, you can't undergo cell division because that's so important to divide your DNA. So that's going to freeze cell division. But other side effects of this is it's not just going to freeze your cancer cell division, it's also going to freeze cell division of your healthy cells. So that's why we have nausea, because your intestinal cells frequently divide. And so if you don't have dividing intestinal cells, it leads to nausea. You lose your hair because your hair follicle cells, which are usually very active, your hair is growing every day, no longer can divide. They can't make more hair for you. And then also you can be more prone to infection because your immune cells, which are so important to your immunity and to your protection, they also divide very frequently. If you stop them from dividing, then you can be more prone to infections. Now, technology is getting a little bit better now. So the specificity of your treatment is improving. So for breast cancer, they've determined if you have a problem with your HER2, then Herceptin works for that. And if you have a problem with your endoplasmic reticulum, then tamoxifen, I think that's how to pronounce it, it's going to be the drug that's going to help best with if that's the cause of your breast cancer. So that's why cancer research is so important, because we want to learn more about these cancers to figure out what targeted treatments we can give to patients that are more effective and not as damaging to their bodies. All right, that brings us to the end of this lecture on 12.3. Hopefully that was helpful to you. Here are the main takeaways. So how does the cell cycle control system work? Okay, hope you can answer that question. How do cyclins and CDKs control the cell cycle's timing? So when do we know it's time to move to the next stage? What is the G0 phase? And just understand the different checkpoints that exist in the cell cycle control system. What signals regulate the cell cycle, tell the cells to continue growing or to stop growing, and then explain cancer. Again, hope that was helpful for you. Thank you so much, and I will see you in class. Bye.